very happy to be here and present this opportunity. Let's directly dive into my PhD topic. So we start with some mandatory bad news. The global population is going to increase and expected to hit like 10 billion by 2050. And this will put a lot of pressure on agricultural systems. We'll need to produce more food than ever before. And on top of that, we are dealing with climate change and its associated challenges such as droughts, increased price pressure, and floods, all of which further threatens the global food security. So we need to find a solution. So based on these points, we desperately need to innovate and breed new crop varieties, the varieties that are resilient towards climate change, but that are also that have an improved resource efficiency. And since there's a GMO skepticism still in the air, the way to go is to breed these crop varieties directly in the fields and field trials. And in the field trials, we are dealing with a large number of varieties and crops. We need to characterize myriads of plots efficiently, and conventional hand measurements can be very time consuming and also labor demanding. And this is where the high throughput field phenotyping comes in. It's a fancy term for non-invasive measurement of crop traits and can be achieved using various platforms. And for the simplicity in this presentation, I'll be just talking about aerial platforms. So, for example, or airborne platforms or spaceborne platforms. We have, for example, satellites, aircrafts, and uncrewed aerial vehicles. And based on our target objective, then we select a platform. In case of breeding trials, it makes more sense to use more small platforms like uncrewed aerial vehicles. They deliver high spatial resolution, so we can capture small plots and usually they cover the area of interest very easily, and also we can fly it whenever we want, unlike the other two options that are also quite expensive. So during my PhD, I focused on uncrewed aerial vehicles, aka UAVs, and once we have the platform, we have to select a sensor, and there are many sensors out there. I'll be talking about only optical sensors. So we have RGB, multispectral, and hyperspectral sensors generally, and again, the choice. There's also a trade-off between them, which one to choose. Do we want high spatial resolution and low spectral resolution, or high spectral resolution and low spatial resolution? Usually RGB cameras have these high spatial resolutions, but they lack spectral resolution, while the hyperspectral sensors are on the other end of the spectrum, while multispectral sensors are actually in the middle. So that's why during my PhD, we have focused on multispectral sensors. They're also quite compared to the hyperspectral sensors, quite cheap. So once we have the sensor, we need to measure vegetation, and vegetation has a very distinct spectral signature when we are measuring them with doing the spectroscopy measurements. And specifically, vegetation absorbs the light in blue and red wavelengths. So this is a chlorophyll absorption. It reflects light in the green wavelengths. This is why the plants appear green. And it also reflects a lot of light in the near infrared region. And based on all of these characteristics, we can use this information to infer the parameters of the vegetation. For example, in the visible range, we can talk about the leaf pigments and retrieve the leaf pigments like chlorophyll content, carotenoid content. While in the near infrared range, we can characterize cannabis architecture. So with this information, we go forward to retrieve the parameters. And there are many parameters out there. We can divide them in two main families. One is biochemical family, which includes the pigments like chlorophyll content, carotenoids, anthocyanins, water content, and the structural family, which includes leaf geometry like size of the leaves, diameter, thickness, maybe, leaf area index, canopy height, and etc. And these are important parameters for the breeders and farmers to improve their management. But how do we retrieve these parameters? There are many different ways how to extract these parameters. So, for example, we have parametric regression methods. They basically are simple mathematical calculation done on the spectrum, and we relate these simple calculations like vegetation indices, as you see here, to the parameter of interest like leaf area index. And then we have non-parametric regression models. These are basically machine learning models that are trained on large amount of data, or spectral data, and also the field data. And then we have RTMs, radiative transfer models, which are physical models, and they simulate the transfer of radiation within from the canopy. And combination of physical models and non-parametric models are combined hybrid methods. And all of these family together can be used to retrieve the crop parameters. So, but how can we choose one? So, for example, the parametric regression models, while they're very simple to use, they are often non-transferable. So once a model is created, it is valid for the taken variety or maybe even sensor, and cannot be easily transferred to another variety or another crop, another sensor. So non-parametric models, machine learning models, they will work well once, as long as a large data set includes our parameter of interest and variety of interest. And if it's not there, then it will struggle. And this brings us to RTMs. Since these are physical models, the solution of RTMs should be transferable to any crop or any sensor because it follows the laws of physics. So this is why we selected RTM for this PhD project. And the RTM that I was working on is called ProCell. It's the simplest of RTMs canopy RTMs out there, and was created some time ago. It was used for satellite remote sensing mostly. It consists of two parts. One is called prospect leaf optical properties model. It consists of variables like pigments, chlorophyll content, carotenoid content, water content, etc. And on the other side, we have canopy optical properties model called a ProCell. And here we have structural canopy parameters like leaf area index, leaf inclination, angle, and etc. And based in the combination of these parameters, we can simulate transmittance and reflectance from the canopy. But since we already have the reflectance acquired from the drone platform, we can actually go to the parameters of interest and invert the model. This is called a model inversion, and basically involves first running the simulations and using these simulations to compare it to the reflectance acquired from the drone platform, and to try to minimize the errors between this simulation and acquired spectra. So once we have the parameters, we have to validate them, and it's done in the field using various devices. In my case, I've been focusing on mainly two parameters, leaf area index and chlorophyll content. And for this reason, we have acquired quite a lot of data. Here you see example of a leaf area scanner, and we have measured various times in various crops throughout the seasons. On the other side, we have the SPAT. This is a measurement device that takes the chlorophyll content measurements, or measure not direct chlorophyll content, but proxy to chlorophyll content. Let's call it that. So we did it throughout 3 years of measurements. And our field trials were located at Campus Klein Altendorf in PhenoRob Central Experiment, containing various crops: winter wheat, summer wheat, soybean, corn, and sugar beet, etc. So this brings me to my research questions. I had two main questions. One of which is how accurately can crop parameters be retrieved using GIAVIS and the ProCell model, because as a ProCell model was created for satellite level data, and the assumptions mentioned in ProCell might not entirely work for high spatial resolution data that GIAVIS deliver. And the second question, we wanted to see if if we can detect and study crop stress using radiative transfer models and multispectral imaging. And to achieve this and answer the questions, we had several projects with accompanying publications. So let's go to each project separately. So we have, first of all, the reflectance calibration. And the reflectance calibration is one of the most important steps before retrieving the crop parameters, because we have to ensure that our image data is good enough. And currently, what we have in the remote sensing community, we have the camera manufacturers offer calibration panels with just one calibration panel with non-reflectance factors, and we take the image of this panel from the shoulder height. But this assumes that when the drone is flying, actually there's no atmosphere between the panel and the drone. While in reality, when we're flying the drone, there's a atmospheric column between the panel and the drone, so the coming reflectance from the panel can be scattered and absorbed by these motor molecules or whatever is in atmosphere before reaching our sensor. So this calibration approach employs a simple relationship. It's called empirical line method. It assumes that the relationship between the radiance and the reflectance of the panel, so the radiance measured from the drone of the panel and the reflectance of the panel itself is linear. But since we have just one reflectance panel, we cannot see if the relationship is really linear or not. And this is why it's recommended to use panels with varying, several panels with varying reflectance factors ranging from dark to light. And this is what we did in this first study. We had panels with varying reflectance factors and compared this approach to the one-point calibration, so one panel approach offered by the camera manufacturer. And here you see some results. We saw some significant discrepancy between the reference reflectance measurements and the single panel approach. And the overestimation, it was especially true for, so we observed a lot of overestimation in the radiation and near-infrared regions, and these are regions that are often used in the vegetation studies. And this was especially true for the near-infrared region. So you see here the results for one-point calibration and overestimation, and here is our approach. So what's outlook here, what we learned? Well, the empirical line method assumes constant lighting, while in reality, while you're flying the drone, even if you're flying for 20 minutes, the lighting conditions are changing. But we assume that these changes are negligible, since we're flying always around the solar noon. What we also didn't include in this study was bidirectional reflectance factor effects. It basically describes how light is reflected by a surface depending on the geometry of both incoming light and the observing angle. But this can be reduced by increasing the side and front overlap of the drone to just focus on the central part of the images. And finally, we have not studied the impact of the flight altitude on the calibration, and this is something that could be done in the future. So to do the similar study was including different flight altitudes. So once we had good reflectance calibration, we are going to the retrieval of crop parameters, here represented by 3 to 4 studies. So I'll just introduce 2 studies in this section. And first, we wanted to see how, if we can retrieve the leaf chlorophyll content and leaf area index by coarsening the resolution of our images, because, as I said, the ProCell model assumes assumptions in ProCell model are valid for the satellite level data, where the data is very coarse, which is not the case in the UAVs. What we did, we actually selected 6 ground sampling distances. So we took our drone data and artificially coarsened the resolutions up to a certain point where we couldn't see the plots anymore. And we did this in the soybean experiment with 2 soybean varieties called Eiko and Mingold. And Eiko is a wild variety with normal green leaves, while Mingold is a chlorophyll deficient mutant, which has a low chlorophyll content in upper leaves and high chlorophyll content in lower leaves. We have collected also the validation data, so measured with SPAT, the chlorophyll content, and leaf area index, and ran the process model to and inverted it to get our parameters of interest. So what we saw was that there was no significant difference between explode resolutions while retrieving the leaf area index. You see here the plot for root mean square error and different resolutions. But what we saw, then we wanted to see how it goes with the chlorophyll content. So first we left the soil and shade in the images, and so that almost no changes in terms of different resolutions. But then we removed shade and soil completely from the scenes by applying some simple thresholding, vegetation mix thresholding. And what we saw was that for one variety, so for ACO, the wild variety, the accuracy of retrieval increased, while for another variety, actually the chlorophyll deficient mutant, it decreased. And this tells us interesting story, a story that of Mingold's two-layer canopy system, with low chlorophyll content in upper leaves and high chlorophyll content in lower leaves. And simply our model doesn't assume that the canopy can have this kind of discrepancy and two-layer system. So what's the outlook here? Well, we established that there was no significant differences between exploratory solutions for LAI retrieval, as long as we can actually see the plot borders more or less. And for leaf chlorophyll content retrieval, removal generally delivers better, removal of soil and shade delivers better results. And this is the advantage of UAVs because we can work on the leaf levels. And finally, Procell assumes a homogeneous canopy, and Mingle has these two distinct layers, and there are models out there called multi-layer canopy reflectance models that can be applied to this case. This brings me to the third publication and third study. It's about the retrieval of crop variables in the maize canopy. Here we wanted to see if the Procell can be actually applied to heterogeneous canopy like maize, because again, the Procell model assumes very homogeneous canopy which is closed. While in case of maize, which has a very complex architecture, we are dealing with rows and inter-row gaps. So usually the Procell model struggles with such cases. So we wanted to see if we can retrieve leaf area index, leaf chlorophyll content, and canopy chlorophyll content, and apply different retrieval schemes. So first of all, in the first retrieval scheme, we left we removed soil and shade from our images and just worked with leaf pixels, and actually applied the inversion to each pixel. So in the end, we had a map of leaf chlorophyll content, and our parameters. In the second approach, we calculate the mean of the whole of the plots and then apply the inversion to this mean to retrieve the crop variables. And this is a long processing pipeline, which includes applying the radiometric calibrations that I first introduced in the first publication, and then do the photogrammetric processing to get to the image. And the second step involves the inversion itself, which tries to minimize the simulated reflectance from the Procell and the measured reflectance. And finally, we applied our schemes. And what we have found out here was that there was no significant differences between different retrieval schemes. So, for example, in mean reflectance approach, we saw, you see here, for LAI, leaf chlorophyll content and canopy chlorophyll content. Generally, the models were struggling to retrieve leaf chlorophyll content, as also has been shown in other studies. And in case of pixel-based approach, so while working in each pixel, we didn't see any improvement in leaf area index and leaf chlorophyll content, but we actually saw quite a lot of improvement in canopy chlorophyll content. And this tells us that actually there's quite some information still hidden behind each pixel. So, what's the outlook here? The outlook is that more complex models, such as 3D radiative transform models, should be explored to assess applicability of high-resolution imagery. And the downside of these models is that they require extensive parameterization and knowledge on these parameters. What one can also do is explore hybrid inversion schemes, which are becoming more of a standard these days. So use the radiative transfer models together with machine learning. And we can also improve the regularization options, so limit the number of bands, constrain the parameters. So there are different ways how to proceed further. And this brings me to the final project project topic, which is a sensor synergy for crop stress detection, represented by two studies, but I'll only focus on one study here. And this study was about multispectral imaging and terrestrial laser scanning for the detection of drought-induced paraheliotropic leaf movement in soybean. Quite a mouthful title. And soybeans are very interesting plants. When exposed to high heat and drought stress, they tend to change their leaf orientation, so leaf angle. So they flip the leaves and also align the leaves parallel to the incoming radiation. And here you see on the top of the second image, actually, the leaf flipping as seen from the drone. The white parts are actually undersides of the leaves of one of the varieties. And here you see how the leaves are becoming vertical, the top leaves becoming vertical, trying to align themselves to the incoming radiation. And this is called leaf paraheliotropism. This is or so-called leaf movement due to the heat and radiation stress. And it's characteristic for mostly the leguminous plant, but as other plant species also have that. But leaf orientation parameters are quite hard to measure, you can imagine. The leaves are moving constantly, and there are many methods out there how to retrieve them. But we wanted to retrieve them with our drone and UAV and spectral, from a spectral data. And what you see here is so-called global sensitivity analysis done on the using our multispectral sensor and the ProCell model. Since one of the input parameters in the ProCell model is a leaf angle distribution, so leaf inclination angle, and this parameter has a very high impact on the reflectance. This is basically what you see here. You are seeing the impact of each variable: leaf chlorophyll content, leaf angle, chlorophyll content, on the reflectance. So we see, for example, for leaf chlorophyll content, it has a largest impact on the in the visible range, while for leaf angle distribution, actually we're seeing that the most sensitive areas are near infrared and red edge regions. But we will measure, we can potentially detect these leaf angle movements from the UAVs, but how can we validate our measurements? And there are different ways how it could be done non-destructively, because leaf angle measurements with handheld devices is very erroneous and also time consuming. So on the one side, we have this stereoscopy, and on the other side, we have the LiDAR measurements. And we decided to go with LiDAR measurements because usually they are much faster and simpler. And this was done in the collaboration with our partners from Institute of Geodesy and Geoinformation. Together with Lina, we went to the field and acquired quite a lot of data. So the research questions were if if we can find the relationship between the leaf angle and the reddish near infrared regions and potentially retrieve the leaf angle from this. And if we can see the effects of the drought using the tertiary laser scanner and the imaging data, because the year where we did the measurements in 2022 was quite hot. And so we had set up the experiment with soybeans again, and Lina's team, they have measured, use a tertiary laser scanner to scan the soybean field, and directly after that, we have flown the drones in the morning and in the afternoon to track the leaf movement. So against these two soybean varieties, and we measured leaf profile content. And I'm not sure what is happening now with these plots because they are not as they're supposed to be. um, okay, well, what we observed first was that there was a high rate of leaf movement for ACO and mingled. So we saw that there was a discrepancy between two varieties. Here it is the data from terrestrial laser scanner. We see that there's a difference between, generally the ACO had a much lower, a much higher leaf angle compared to mingled. And oh my, okay, something is wrong with my slides, but I'm not sure what, because I had, okay, whatever. And we also observed that the change of leaf angle was much pronounced in ACOs, so wild variety compared to mingled. And especially on the last date, so this was in August, we see that rate of change was for ACO was much larger, and we think that this is attributed to mingled's. Mingled is not changing leaf angle because it has low chlorophyll content in the upper leaf, so it actually reflects more sunlight back in the atmosphere. So there's less thermal radiative load on the leaf, but this should be studied later with more precise leaf level measurements. And we're sure that there was drought because there was an unusually warm and dry summer in 2022. And you see here's reference measurements and how they align with the soil water content in the study area. Then we compared our correlations with, our measurements with the spectra, and what we found out was that there was no correlation in the morning between the selected wavelengths of 740 and 842. So we studied first, so we wanted to see if there was also a relationship between leaf area index and leaf angle and between the selected spectrum, because these two varieties are actually sharing the sensitivity in the Procell model. But we found out that there was definitely the positive correlation for the leaf angle for the red edge and for the near infrared when both of varieties are combined. While for leaf area index, there was no correlation in the afternoon. So, but we cannot just work with one time of measurement. We wanted to compare both morning and afternoon measurements, and basically to normalize these measurements, we can use vegetation indices. And what we did, we just calculated simple ratio index, divided 740 by 840, and indeed here we started to see quite a good correlation. The size of each blob actually represents the leaf area index, so the larger the point, the larger the leaf area index, and we see here that the relationship is not governed by the leaf area index because different leaf area indexes are all over the place. So what we did next, we tried to remove the soil to see how it affects the retrieval. We saw that the accuracy increased, and then removed the shade. and it increased even further. So what's the outlook here? The outlook is that more research is needed in this direction. The leaf area index, leaf angle distribution is a overseen parameter, even though it's one of the main parameters. And what could be done is to explore different crops which have exhibit paraheliotropism and different sensors, and to expand the diurnal measurements. And we also had the FUSE publication, which I will not talk about here. It was a large collaboration with different partners from various institutions, where we wanted to create a guideline how to use different sensors to detect the crop stress and guide the newcomers to this field to use these sensors to measure the parameters. So what's the summary? The summary is that high-resolution UV imagery, when integrated with ProCell, actually yield accurate results compared to satellite level studies. And particularly for variables like leaf pigments and in homogeneous canopies, we can retrieve these pigments with UVs, which wasn't the case with coarse resolution data. And for the first time, we demonstrated the ability to detect the paraheliotropic leaf movement using the sensor synergies. Our findings confirm the ProCell simulation, and I hope in the future it will be used more. And finally, I wanted to say that radiative transfer models are have been neglected in the community for too long, and I hope that this study will encourage a greater visibility and application of these models. And with that, I'm finished with the talk. Thank you very much.